Hello and welcome everyone to this podcast. In this podcast we're going to talk about how mutations form, and we're going to talk about spontaneous and induced mutations. Your book is going to go into fine detail about some of these mutations, but what you really need to know is what I'm going to tell you in this podcast. We're going to try to break it down for you to exactly what you need to know. The first thing is you need to know the difference between a spontaneous and an induced mutation. So spontaneous mutations. With spontaneous mutations, as the name implies, are mutations that form naturally. They could be mistakes in replication, DNA replication. Or we could have chemical changes to the DNA, which of course then get replicated erroneously. And these chemical changes to the DNA are not caused by any outside chemicals being presented. These are changes that just naturally occur to the DNA, specifically to the bases. And we'll talk about those first. The next thing we'll talk about are induced mutations. And as the name implies, these are mutations that form because of some sort of outside agent acting upon the DNA itself. So these are environmental changes to the DNA sequence. Okay, we're going to first talk about spontaneous mutations. And when we think about spontaneous mutations, we think about ways that the nucleotides are changed naturally, not by some induced chemical state. And the first way we're going to talk about it is through a process called depurination. As the name appears, this is a process where a purine is lost. So a guanine or an adenine is lost. Alright, so let's draw out what depurination might look like at the DNA level. And on one strand we're going to put C-A-T C-G. And then we're going to have the complementary base pairs here, G, T, A. Then a G, and then a C. In the process of depurination, what happens is we're going to lose this guanine. So we'll start off our CG here, our AT here, TA, and then this is where we're going to have an error. We've lost that purine guanine. Now, fortunately, usually what happens is this is repaired and a G is placed back in here before it's replicated. However, sometimes it's not fixed. Now, if it's not fixed, what happens? Well, the problem comes when this begins to be replicated. This half here on your left is replicated fine, because remember, through semi-conservative replication, we use each half as a template. When this is used as a template, no problems at all. When this half is used as a template, either we'll skip this one, and now the new DNA will have one less base, or a random nucleotide will be put in there. In either case, this is going to create a mutant allele here. And this is all because we lost this purine and weren't able to fix it. Depurination is loss of a purine, and then an error in replication. Okay, that's all I want to say about depurination. The other way bases can be changed that we want to talk about is called deamination. Deamination. This is what you need to know about deaminations. This is when a base loses an amino group. This is most often seen when you take a cytosine and convert it to a uracil by losing an amino group. Or you can take a cytosine and convert it to a thymine. Technically, to go from a cytosine to a thymine, there's one other thing that has to happen. But for our purposes, know that a deamination can convert a cytosine to a uracil or a thymine by the process of losing an amino group. And now that changes that one nucleotide and can lead to a missense mutation or a nonsense mutation. The next thing I want to talk about with spontaneous mutations is the role that reactive oxygen species has in the cell. Now, we just are going to abbreviate it as ROS. This is the product of oxygen metabolism. We make a lot of this in our cells all the time. Luckily, we have enzymes that scavenge these oxygen radicals. So we should also say that sometimes we refer to these as oxygen radicals. Again, we have enzymes that scavenge these and remove these oxygen radicals, which is good. But the ones that don't get scavenged, or if you make too many of them, they can begin to damage the nucleotides. And they can damage the nucleotides by oxidizing nucleotides. I'm going to erase this to give us a little bit more room to talk about. Probably the best known example here is guanine. Guanine, when you expose it to reactive oxygen species, it makes a modified guanine. And we're going to call this G with an asterisk by it. And over here, we'll just call this G as we've always done. Now, in a particular DNA sequence, you may have a G binding with a cytosine, like you should. Now, the modified guanine doesn't bind with cytosine. Instead, it binds with adenine. Now, during replication, what's going to happen is this modified guanine is going to continue to make a base pair with adenine, because that's what it recognizes. And the adenine, when it makes new DNA, is going to base pair with thymine. As you can see, we're changing the nucleotide, and by doing so, we're going to change the DNA sequence, and if it happens to land in the appropriate genes, it's going to cause a missense mutation, a nonsense mutation, a variety of mutations that we talked about on the first podcast. The last thing I want to talk about, and I'm going to talk about it in very general terms, are these trinucleotide repeats. Trinucleotide repeats add, as far as the DNA is concerned, it will add a series of 3 nucleotides, and they're repetitive. For instance, a classic one is CAG. And by adding several of these CAGs, what you end up doing is adding a whole bunch of glutamine amino acids. Glutamine is the amino acid coded by the codon CAG. And having a whole bunch of these glutamines, particularly at the end of the protein, can cause damage to these the ultimate protein. Now, there are several diseases linked to this, but a classic one is Huntington's disease. So all I want you to know about trinucleotide repeats is that it's the addition of 3 nucleotides in a row, and it's the same. And in this particular case, and you should know this case, it's a good one to know, it adds a series of glutamines that can lead to the disease Huntington's. Alright, let's go ahead and continue our discussion, and now let's start focusing on induced mutations. As we said before, an induced mutation is one that is caused by some sort of environmental or external factor that changes the sequence of a DNA. So critical to the understanding of induced mutations, we have to talk about this term here, and that is a mutagen. What is a mutagen? Well, a mutagen is that agent, that external or environmental factor, but we'll just call it an agent. Agents that alter the sequence of DNA. And this is an external factor. It's nothing generated internally. It's an external factor. And we're going to talk about 3 such factors. We're going to talk about ones that cause base modification. And then we'll talk about base analogs. And we talked a little bit about base analogs when we talked about treatments for HIV. And then we're going to talk about UV and X-rays. Various kinds of ionizing and non-ionizing radiation. Okay, let's start talking about modifications, base modifications. Base modification. These are going to be agents that, these are mutagens that alter the structure of a base. Similarly, when we talked about depurination and deamination, the difference here is though that this is caused by an outside agent. One such base modifier I want you to know about are alkylating agents. And what I want you to know about these alkylating agents is that they transfer an alkyl group to DNA. More specifically, to a base in DNA. And what this will do is cause mispairing of DNA during replication. And you should also know that many poisons work this way. One that is associated with World War I is mustard gas. Mustard gas is an alkylating agent, and it can damage the DNA and lead to long-term mutations that can have downstream consequences to the individual. But as a take-home, know that alkylating agents transfers an alkyl group to DNA, and this can cause mispairing of DNA during replication, which then passes on that mutation in every subsequent round of replication. And now I want to talk about base analogs. And I'm going to talk about one example here called 5-bromo uracil. So this is a uracil with a bromine attached to it. If this base analog is incorporated in the cell during replication, it can sometimes use 5-bromo uracil in place of thymine. So we're going to put here that it looks like thymine. And so sometimes the cell will take this up and use it. Okay, so what's going to happen here is, let's go ahead and draw our DNA. And we'll just draw some random bases here. They don't matter so much. But what I want to talk about is this one right here, the adenine and thymine. During replication, what can occur is that the thymine can be replaced with 5-bromo uracil. And now the problem will come during replication, when these strands are separated. So this strand here, I'm going to draw them smaller up here just so I can put them both here. We'll have the A over here, right? This strand. It'll incorporate a T, so that one's fine, no problem there. Now this one, on the other hand, we have the BU here, the 5-bromo uracil. It will form a base pair with guanine. So now we've introduced a guanine here. And now that's going to change the sequence, of course. And so any genes that this happens to be in, instead of having an A there, will now have a G. And again, now when this goes through division, all subsequent daughter strands are going to have a guanine here instead of an adenine. Over time, this will become much more predominant in the cells within that tissue. And if it turns out to have a negative consequence, that can cause damage to the individual, of course. Alright, so this is what a base analog does. It replaces one of the existing nucleotides, and because its structure is a little bit different, it's going to base pair with a different nucleotide. Now the last thing I want to talk about with induced mutations are X-rays and UV light. X-rays. I'm not going to spend a lot of time talking about X-rays, but X-rays are deep penetrating ionizing radiation. So we can say ionizing radiation. These X-rays can lead to the buildup of reactive oxygen species, and that will go on to affect the DNA just like it did when it's internally produced, the reactive oxygen species. The last thing I want to talk about are UV light. UV light will lead to what is known as thymine dimers. So if you have in a strand of DNA here, if you have two adjacent thymines, what can happen is these thymines will, what we say, dimerize. They are connected. These thymine dimers cause two problems. They cause problems in transcription, TXN for short, and replication. These dimers interfere with the replication machinery to copy these pieces of DNA, and the transcription machinery cannot read through these to make the appropriate messenger RNAs. And that's all you need to know about UV light and the way it can form thymine dimers, and then how those thymine dimers affect transcription and replication. That's all I have for this podcast on spontaneous and induced mutations. If you have any questions at all, please make sure you come see me, and I'll help in any way I can. I'll see you in class. Bye.